Now we'll see dark reaction, which is biosynthetic phase. Here, dark reaction means, it doesn't mean it will happen, the process will be in the dark. Light energy is not necessary for the dark reaction. That is the thing. But indirectly it is depend on the light reaction. That I shown here. ATP and NADPH are produced in the light reaction, and that whatever the products came from the light reaction will combine with the carbon dioxide and water in the dark reaction and forms the sugars. So sugar formation, where it will be taken place in the dark reaction by using the products of the whatever the light reaction. If the products are not there, there is no dark reaction. That only he has given one paragraph. That is where it will be used. NADP and ATP. ATP and NADP will be used in the dark reaction. And a dark reaction, after completing the light reaction also, as long as ATP and NADPH is available, which are synthesized in the light reaction, the dark reaction will be continued. If they are not available, this will be stopped. And in the starting one video when I told, in the C6H12O6, C6 and O6 will come from the carbon dioxide, hydrogen will come from the water. That water, how it is coming? It is bringing by the light reaction in the form of NADPH. That hydrogen will be joined with the carbon dioxide and it will form the sugar. That only I shown here. So light is not needed in the dark reaction, but needed light reaction products, that is ATP and NADPH. Now we will see that carbon dioxide in this reaction, the dark reaction, carbon dioxide combines with water and forms the sugar. This is the two, carbon dioxide and water. It will combine with the help of the products which are ATP and NADPH, which was produced in the light reaction. And this is carbon dioxide with the forms of sugars. This is the discovered first by the Kelvin. Kelvin is the scientist. In the Second World War, they came to know the importance of the radioactive elements. So the radioactive elements is by using that C14 radioactive isotope, he find out in this dark reaction which stable product is to rise. That stable product is nothing. That is the 3-phosphoglycerate. So 3-phospho means third position carbon. The phosphate is attached, 3-phosphoglycerate. And remember one thing, glycerate and glyceric acid both are the same. One is the glycerate means a little bit salt it is. And glyceric acid means in the dissolved condition. There is no difference. So phosphoglyceric acid will form, and this is the three carbon compound. So they are called as C3 plants. First stable product which is produced in the dark reaction by taking the carbon dioxide is 3 carbon compound. So it is called as C3 cycle also. This is important. And next in this one, later on the Kelvin has discovered the entire, all the pathway, how it will travel, that is the 3-phosphoglyceric acid, how it will pass and how it will take the ATP and NADPH and how it will form the sugars. He has done the entire thing, he has completed. But the thing is, when C3 that's the 3 carbon compound is generated first by, he worked on the algae, this also little bit, and found. And next, the C3 plants are, the C3 type of Kelvin cycle must be done by all the photosynthetic plants. Means photosynthetic plants and anything, C3 is common. When he found that one, other scientists, they tried to find out all the plants will have the 3 cycle. First carbon dioxide fixation compound is the 3-phosphoglyceric acid only. Then they found some of the plants are shown. First carbon stable compound is C4, 4 carbon compound, that is oxaloacetic acid, OAA, oxaloacetic acid. So that group of the plants are called as C4 plants. Now we will see the C3 cycle, which must be, the C4 plants also has to do the C3 cycle for the producing of the sugars. Remember this point? I will cover that one in the C4 plants. Now, how he has done the Kelvin? By using the radioactive isotope C14, and next will be the primary electron acceptor. He has given one sub chapter. What is primary electron acceptor? The Kelvin only discovered primary electron acceptor is 5 carbon compound, that the ketose sugar, ribulose bisphosphate. Ribulose bisphosphate is the first carbon dioxide acceptor in the Kelvin cycle. Then up to that time, he discovered what the scientists are thinking and doing the research, that the first stable compound is the 3 carbon compound, that is phosphoglyceric acid. So this may be primary electron carbon dioxide acceptor, may be 2 carbon compound. They are trying to find out, but all of a sudden they came to know it is not the 2 carbon compound, it is a 5 carbon compound, that is the RUBP, ribulose bisphosphate. So RUBP is the first acceptor of the carbon dioxide along with water it will take, and the enzyme name is, which is the RUBP, accept the carbon dioxide and water in the presence of ribosco. This is called as, the enzyme is RUBP, ribosco. Ribosco means ribulose bisphosphate carboxylase oxygenase, that enzyme name. This is the only enzyme which can take the carbon dioxide and oxygen also. Generally, the active sites will take only one type of the substrate only. Here the substrate may be the carbon dioxide, or maybe it is the oxygen. So oxygenase, carboxylase, we are calling that also he has given. Now the thing is, the Kelvin cycle, all photosynthetic plants do this because the sugar preparation is the final product of the photosynthesis that will be done in the Kelvin through the Kelvin cycle in the dark reaction. When we are seeing the Kelvin cycle is generally it is having the 3 steps. The main 3 steps it will be completed. One first step is the carboxylation, means carbon dioxide fixation, and the carbon dioxide fixation will be reduced and forms the sugar, and the ribosco, the ribulose bisphosphate also will be regenerated in the cycle. Out of these 3, first we will see the carboxylation. The carboxylation, whenever we are seeing the carboxylation, that is the ribosco or RUBP, 5 carbon compound, by taking the carbon dioxide and water with the help of the ribosco enzyme that I told already, it will form this is called as carboxylation, and it will form the 3 carbon compounds. Already it is 5 carbon, it is 1 carbon, it must form 6 carbon, but 6 carbon compound is unstable. So it will be splitted into 3 carbon compounds as 3-phosphoglycerate or glyceric acid. Now, D-phosphoglyceric acid will take the ATP and NADPH and reduces the carbon dioxide into the triose phosphate. Means this will be added, that ATP will be used as the energy and NADPH will donate hydrogen to the carbon dioxide and becomes the NADP plus, and then the carbon dioxide will be mixed with the hydrogen and forms a triose phosphate, which we are calling it as the G3P. This also triose phosphate is also called as glyceroldehyde 3-phosphate, which comes in the glycolysis of the respiration. That is different, there I will discuss. And this is also called as a phosphoglyceroldehyde, PGAL. Now the thing is, after this thing, whenever it comes, G3P, triose phosphate is formed, 1/6 of the triose phosphate will be sent to formation of the sugar, and next will be remaining will be sent to the formation of the RUBP. So here the regeneration of the all the 5/6 of the triose phosphate will be regenerated into the RUBP by taking one more ATP. This he has given in the textbook, but the thing is, you may get the doubt why the ATP is required. Here 1/6 has gone out, then what happens? 1/6 has gone out of here 5 carbon, added 1 carbon, and 2-3 carbon molecules for phosphoglycerate has formed, which are having the 6 carbons in the both, and 1/6 has gone, 1 carbon has gone. Now how many carbons are there? 5 in the form of the 2 carbon and 3 carbon. They will be united and forms the 5 carbon compound. Uniting the both the things required energy, that is this energy. Remember it. Why it is taking one more ATP for the regeneration means the 2 carbon and 3 carbon both will be mixed together in anabolic reaction. So it required energy. So it is taking. Now, in the taking of this one, total in the cycle, whenever we are observing regeneration also we saw, out of this one, most crucial step is carbon dioxide fixation. And next crucial step is regeneration. Because the regeneration is not there, then again RUBP is not as it has to cell size again. So, regeneration is also crucial. Reduction is common, it is to take place. So, on the overall, whenever we are seeing, here it is that 3-phosphoglycerate, whatever is there, that 1 carbon is coming. Now you observe this one, carbon is adding to the 5 carbon compound and dropping in the triose phosphate formation for the sugars. All the 5 carbons are going and forming again this one. This is the cycle. And now if you want to observe very clearly, we take 6 RUBP and it will take 6 carbons and forms, already 2 are formed, 12 are formed, and ATP 12 molecules will be taken, and NADP again 12 will be taken, because 2 ATP molecules are taking in the not here, here only 12 ATP and 12 NADPH are taking, because 6 molecules are present, 6 carbon, 2-3 carbon compounds, and so it is 12 molecules will take 2 each, not this one. Anyhow, this is not necessary to explain. Now the thing is, the regeneration, why the ATP is required means 3 plus 2 carbon compound has to mix together and form this one. In this way, 2 for 1 molecule, 1 carbon fixation require 2 ATP and 2 NADPH for 1 And now this one molecule and regeneration, it required one more ATP. So 6 rounds it is taking. That it will be the one carbon, like 6 carbons will come and it will form the sugar C6H12O6, that is the glucose. Glucose will form the starch, glucose will form the sucrose, everything. Now, how many ATPs will be taken for the formation of one glucose molecule? That one ATP molecule produced means here 2 ATP and here 1, so 3 ATP it will take. Such type of sugar carbon molecules, one cycle required 3. 6 cycles, 6 into 3, 18 are required, that he has given in the last. And how many NADPH are required? It also given in the last, because NADP will be used only one place, that is the 2 NADP for one cycle, and C6H12O6, it is the 2 into 6, it will be the 12 only. So 12 NADPH are required and 18 ATP are required for the formation of one glucose molecule. Now, if you go to the back, one more paragraph he has mentioned, in the cyclic and non-cyclic electron transport, non-cyclic will produce ATP and NADPH equally. But here ATP requirement is more. How it will reach that purpose? For that only, cyclic is also in the stromal amylase, cyclic will be taken place, which is to produce only ATP. The extra ATP which are produced in the cyclic and non-cyclic ATP and NADPH is enough to complete all the thing. This is the dark reaction, how the sugars will be produced. And this thing he has not given clearly, very clearly in the textbook, but the thing is, you read it and understand. If you are having any doubt, please call me, my number is on Tamil. And you can contact and send your message also. If you like this video, please subscribe my channel and put it the like. Thank you.